So, good afternoon, everybody. On behalf of the UK Chemical Network, on behalf of Glenys Cutting, Simon O'Connor, and myself, Christian Masad, we welcome you to the second of the UK Chemistry Network meetings. It's a virtual meeting, and our speaker this afternoon is Tristram Wyatt, who is an evolutionary biologist at the University of Oxford. He has a particular interest in pheromones across the animal kingdom, including humans. So we welcome Tristram today.
Thank you very much, everybody, for coming. So I'm going to be talking about the human sense of smell and human pheromones. And if you'd like a copy of the slides, just send me an email, or they'll be going up online after this talk at the address there. So the thing which brought me into thinking about humans was writing the book down in the bottom left-hand corner, Pheromones in Animal Behavior. And the last chapter is all about humans. But along the way, I'm going to be talking about power-posing politicians, and we'll be returning to those in a little while. So this is the plan for the talk. I'm going to start briefly with talking about the human sense of smell, and then pheromones in general, what we expect from pheromones and how we find them. Talk about human pheromones, and that leads us into the question of the reproducibility crisis, as it's been called, in the life sciences. And one of the impressive things about psychologists is the way that they have been approaching how to do it better, how to improve the science. And I think there's a lot that we, looking at the human sense of smell and pheromones, could learn from that. So what about the human sense of smell? Well, speaking with the audience I have today, you all know how important the sense of smell is. We can go back to early reviews by Dick Doty, an important one by Schall and Porter, the recent paper by McGann in 2017, and then a whole symposium devoted to olfactory communication in humans, which is available free online. It came out in April, coming from a conference held in 2019. So we know that the sense of smell is very important to humans and might even be involved in communication. But one of the things about the human sense of smell is that our studies, often looking at subtle effects, that are done in a very small scale kind of way. And many of the techniques come from psychology. Now, the thing that we can say about small scale experiments in psychology is that many of them turn out to be hard to replicate. But there are new opportunities to do large-scale studies. And one of these comes from something I'll be mentioning later, the Global Consortium for Chemosensory Research. And of course, the sense of smell is very much more important than we realized previously, because it's coming up as a symptom if you have a sudden anosmia in connection with COVID-19. Now, we've known about pheromones for a long time, but it was only in 1959 that the first one was identified. And this wing fluttering by the male was the crucial observation that allowed the chemist to track the presence of the pheromone as he made his fractionations. So this was the first identification of a pheromone, and in doing this, Budendant the chemist established the gold standard for identifying a pheromone. So he started with the bioassay, the wing fluttering of the male in response to the presence of the female pheromone. He then isolated from 500,000 female moths. He made a very wise choice. Having fractionated and concentrated, he was able to identify the molecule concerned, he synthesized it, and then crucially, he went back to the male moths to test in the same bioassay that he had indeed identified and synthesized the correct molecule. And as you'll know, this is still basically the essential set of steps for identifying a pheromone. Now, since 1959, pheromones have been found right the way across the animal kingdom. And what we'll see is that your favorite animals are there, along with birds, perhaps, in fact, highly likely, and also a reminder that it works just as well underwater in animals such as goldfish and crabs. But what about human pheromones? Why would we expect pheromones in humans? And the reason is simply because we're mammals. Our smells change at puberty, and that's because the apocrine and sebaceous glands start to get active, and they are activated along with the axillary and pubic hair. So with any other mammal, we'd be looking for pheromones. Now, in terms of humans, there have been 3 waves. In the early 1970s, there was a set of papers by Michael, and others on copulins found in the vaginas of female rhesus monkeys. In the 1970s and 1980s, the pig pheromones came to the fore, mostly because you could buy them in an aerosol can. But since 1991 through to the present, androstadienone and estratetraenol have taken over as the molecules that are the subject of most studies. So this is the 3rd wave from 1991 to the present, and sadly, it's a case study of how not to establish the identity of a human pheromone. It seems that the originators simply chose 2 molecules, claimed they were pheromones, but provided no evidence. And I've described the story in 2 papers, one earlier out this year in April 2020, and one from 2015 that goes a bit deeper into the history. So these are 2 fairly obscure steroids. Earlier this year, I did a scholar search, and you can see that up to 1990, there really is very little interest or citation interest in these 2 molecules. But from 1991 through to the present, there's a lot of interest, more than 1,000 papers in the case of androstadienone. So what happened in 1991? Of course, it was this paper, which you may be familiar with. It's Monty Blanc and Cross, and it's the effect of putative pheromones on the electrical activity of the human VNO and olfactory epithelium. These were the 2 molecules, and it's important to note that they were supplied and patented by the Erox Corporation, set up by enterprising professors at the University of Utah. And they had the aim of selling these as human pheromones to be added to fragrances. But it really is a mystery why those 2 molecules were chosen. There were plenty of other candidates. If you take swabs from a human armpit and run them through the GC, you'll find hundreds of compounds, going probably into the thousands. So what had led them to choose these 2 molecules? Had they done the Brutendant gold standard approach of systematically searching for the molecules? And the answer, of course, is no. None of the steps were ever carried out. And we don't even know where they collected their samples from. There was a lot wrong with the paper in 1991. It simply wasn't a paper that could have established that even putative pheromones exist. It wasn't that comprehensive testing of all the human secretions. It was just 5 molecules that came from the corporation. More problematic, it relied on testing the VNO, which doesn't function in adult humans. So physiologically, it was something of a nonsense. The sample was very small, but they did get a positive result. I think nothing might have happened except that it was endorsed by a leading scientist in the field, Professor McClintock. And this was the paper that came out in 2000. They used those two molecules despite the lack of evidence for efficacy. And the only reason for using those two molecules was the 1991 paper. But they were commendably cautious, commenting in their paper that it was premature to call these steroids human pheromones. Not everybody else that's followed has been quite as cautious. So by the beginning of this year, there were some 61 papers using one or both of these molecules, and almost all of them were reporting positive effects. There were a variety of physiological and psychological effects, and these were coming from many good scientists, and they were often published in prestigious journals. And these studies get everywhere. That's why there are so many citations when you search for the molecules. But there's no more evidence now than in 1991 that there is anything behind the molecules other than the claim in that original and rather flawed paper. So how can we explain the results and a lot of papers? Well, one possibility is that even if they're not pheromones, these two molecules could have effects, and that's certainly possible. But that could equally be true for any of the 700 or more molecules given off by humans, which have never been tested. And who knows, those molecules might have even greater effect. Now, there is an alternative explanation that these are probably false positives, but so many, it really does cause a bit of a problem. So could so many studies be false positives? And are there any precedents? Well, the answer is yes. And I'm going to give you just three from cancer research, from a serotonin receptor, and depression, and power posing. And this all relates to the reproducibility crisis in the life sciences. And the problem is that you can build a complete scientific field from an initial influential false positive. So this is from Begley and Ellis in 2012. They were reporting an attempt to replicate findings in cancer research. And what they noted is that hundreds of secondary publications could expand on the elements of an original unreproducible observation. But none of these secondary publications actually sought to confirm or falsify the fundamental basis. They took it as a given and used that as the basis for their own study. In research in depression, it's actually a serotonin transporter gene, not a receptor. And a 1996 study suggested that there was an association between this particular variant and depression. 450 papers built up. There were multiple meta-analyses, but actually there was no underlying association when it was investigated in a very large sample in 2019. And that brings us to power posing. So these are three Conservative Party ministers. The two on the left are from a Conservative Party conference. And what seems to be the case is that their speech coach had watched the second most viewed TED Talk ever. And this is one on power posing, that currently it's at more than 57 million views. And it's a very attractive idea. So it seems to be true that we display our confidence by our posture, how we walk into a room. And Professor Cuddy, a professor at the Harvard Business School, had the very interesting idea that could the reverse effect be true? If you posed somebody's body into a confident pose, would that affect our physiology, our behavior, and change the way we behave? And so the paper was published in Psychological Science in 2010. So the basis of the experiment was that students were assigned randomly to be put into high power poses. So the student volunteer, was actually placed in position in one of these high powered poses, as called, standing up, legs apart, hands behind head, or they were put into a low power pose, and again, beautifully randomized. And then various measures were taken. So immediately afterwards, they were given a test that investigated whether people would take more or less risk, and a salivary testosterone and cortisol test was taken. And the answer from n equals 42 in the original paper was yes. The high power poses had significant effects on risk-taking and on these hormones. So the idea was very attractive, and over the succeeding years, there were some 50 studies inspired by the idea. And these are what we call conceptual replications. These were like the cancer studies. They didn't test the original idea in a proper replication. They were inspired by the idea and tried in a different context to see whether they could come up with something similar. But when finally 5 years later, in 2015, a true replication was tried with a very much larger number of participants, it failed to replicate. Now, people did feel more confident, but there were no effects on the important effects as described in the original study on risk-taking or the hormones. And I describe in more detail what was going on in my 2020 review. So there was a major problem. It simply failed to replicate. But the dispute ran on for many more years. And I'll return to this particular study to see how it was resolved finally. Now, you may be wondering, this was all in Psychological Science. What about a really top journal, in inverted commas? And the answer is yes, these problems do go right the way through science. So Camara et al. Replicated the 21 social science experiments they could find that had been published in Nature and Science in the period 2010 to 2015. And the papers had titles like these: Avoiding Overhead Aversion in Charity. If you're told that your donation will go all to the good cause because a foundation has covered the overheads of your chosen charity, it does look as though you give more. And there's another example here: Spontaneous Giving and Calculated Greed, which actually I think did not replicate. Now, in the replications, they increased the sample sizes dramatically, and usually were able to involve the original experimenters to advise them on the replications. But for the results, only 62% the studies could be replicated with a significant effect in the same direction as the original. And even in these, the effect size was halved. But there was another twist to the study. In addition to the replications, they recruited more than 200 volunteer scientists who were given an imaginary $100 each to bet on which studies they thought would be most likely to replicate on the basis of reading the paper concerned. And the results were surprising. In fact, it was almost a straight line with the results from the replications. it was possible to predict with a high level of accuracy which studies would replicate and how well. And that leaves us with the question, why didn't the referees and editors spot this too? And of course, the answer is that all of us do like a good story. And if it's persuasively written, we are likely to agree, because if it makes a good story, it's very attractive. And incidentally, subsequent studies using amateur volunteers in a Dutch study proved that they, as volunteers, could spot whether papers were likely to replicate or not. And this led the science journalist Ed Yong to waggishly suggest that the only sure thing in psychology now is that only 50% of studies will replicate. So what's going on? We've got exciting research that won't replicate, but what's very impressive is the way that psychologists are analysing the problem, and have been over the last decade, and suggesting solutions that might really change things. And they've also identified a key problem. We really need, or have the feeling, that we must publish in high-impact journals for survival, if nothing else. And there's a very nice one-page synthesis from Dorothy Bishop that came out in 2019, and she's describing the 4 horsemen of reproducibility. And she points out that many researchers persist in working in a way almost guaranteed not to deliver meaningful results. They ride with what I refer to as the 4 horsemen of the reproducibility apocalypse. So we have the problem of publication bias, that if you get a negative result, it goes in the file drawer and never gets published. There's the problem of low statistical power, very small experiments, and very little effect. There's all the problem of p-value hacking, which I'll come on to, and harking, the hypothesizing after results are known, which, if you're looking backwards, it's almost like drawing drawing a ring around the arrow that's already landed, and it's very hard to miss. And what really seems to be causing the problem is the unlimited researcher degrees of freedom. It's not malicious intent. And this was brought out very nicely in a paper from 2011 called False Positive Psychology. Undisclosed flexibility in data collection and analysis allows the presenting anything significant. And what they point out is that the researcher has so many decisions to make: to collect more data, to exclude some observations perhaps or not, which conditions to combine or compare, what the control variables should be. And these are just the starting points. There are so many things that can be decided in data collection, the analysis, but finally in the reporting of an experiment. And given that flexibility, there's a great increase in the false positive rate that you get. And to illustrate this, they conducted an experiment with real students, which showed something surprising: that listening to the Beatles song When I'm 64 will take a significant number of years off your age. So they produced a summary, and in bold and highlighted is what they reported. In grey text is the full details of what they really did. And you can see there's a lot missing in the final report. The number of students reported in the final numbers has gone down from 34 to 20. Actually, they had 3 songs, not 2. And they've used father's age to control for variation in baseline age across participants. But they don't tell you that if they didn't do that, all the significance disappeared. And it's a fun paper with lots of advice to referees and editors about how to avoid some of the problems. The point they're making is that doing experiments and analysing them in the way that so many psychologists had come to find perfectly reasonable actually meant that very unreliable research would get published. And this led them to an observation in 2018 that the file drawer effect was actually much worse than a file draw. And what they suggest is that with so much p-hacking probably going on, without people really realizing that what they were doing was p-hacking, is that researchers were trying out many analyses, but only reporting the ones that were statistically significant. And it actually explains a paradox. The majority of published findings in psychology are statistically significant, but the majority of studies are underpowered, which should mean that significant results are rare. So what's going on? And they suggest the explanation is it's the failed analyses, not the studies as a whole, that go in the file drawers. And the disturbing conclusion they come to is that most failed studies are not missing. They are published in our journals masquerading as successes. So the results that looked as though they weren't significant actually are massaged in unwitting ways until the results are interesting successes. So the problem, as previously I've mentioned, is what we're looking for and what we prioritize is novel, eye-catching and exciting results. And then we target these at journals with a high impact factor. What this gives is a strategy that's optimal for career advancement, but it's not good for science. There's a real conflict. And of course, there's no funding or incentive for replications. And while this differs between fields, so in physics, replications are relatively common, in the life sciences, they are extremely rare. And these ideas are brought together in a great paper, a manifesto for reproducible science that came out in Nature Human Behavior in 2017. So what I want to move on to for the final third of the talk is how can we do it better, and how can we be inspired by psychology's renaissance? It's been brought together at book length by Chris Chambers. It's the 7 Deadly Sins of Psychology. Each chapter is devoted to a particular sin, but he also provides suggested solutions to make things better. If you want it in 3 or 4 pages, the paper I've just mentioned provides that. Chris Chambers is again one of the authors, along with Dorothy Bishop. And Nelson, Simmons and Simpson provide an optimistic view of psychology's renaissance, also providing a good account of how we got to where we are. And there are 4 references in the 2020 paper that I've mentioned. So what are some of the take-homes from Chris Chambers? Well, these include pre-registration of experiments, and I'll come on to that in just a moment. The idea of registered reports with pre-acceptance of papers before the results are obtained. There's a strong recommendation for consortium research coming together to get much more powerful experiments. And that is combined with open science, with data and code sharing, open access publication, and crucially, moving on from journal impact factors. And what we ought to be rewarding, what we ought to be doing is changing the culture so that we reward reliability and reproducibility, not excitement, not simply novelty. And I'm pleased to say that within the area of chemosensory research, the Global Consortium that formed earlier this year and now has more than 500 scientists from 55 countries working together, and they're using pre-registration, they're using open science techniques, and again, they're using preprints. And the message is, do join us if you're interested. So what about pre-registration? This is all about removing the temptation to look at your results on your methods flexibly while you were doing the experiments, or even worse, afterwards. So before you start the experiments, you declare the full methods, including the analysis, and there are various tools. You could just put it in an envelope, but these allow you to share these after an embargo if you wish, and this allows you to share with your collaborators what you're intending to do. And there's a good account of these in the pre-registration revolution by Nosek et al. You can take it a stage further with pre-registration, which then also goes to peer review, and that can lead to registered reports. So this is where you have peer review before the results are known. So you have your idea, you design your study, and this is stage one. It's sent out to the journal, the editor sends it out for peer review, the reviewers assess whether the research question is important, whether you have a large enough sample size, whether your methods are going to be sufficiently rigorous, and if they are, you get an in-principle acceptance. You then go to collect the data, and you submit your stage 2 manuscript, which now contains the data. The reviewers assess the compliance with what you said you'd do, and whether the conclusions are a reasonable set of conclusions based on the evidence, and then they publish the report. Now, the important thing here is that so long as you do what you said you'd do, your paper is guaranteed to be accepted, whether or not you find something exciting. So in other words, a null result is still an interesting publication. You've actually made a contribution to our knowledge. Daniel Simmons, one of those authors you've seen in a number of papers, reminds us that it eliminates the bias against negative results, because when the study is reviewed, those results are not known. And Chris Chambers suggests that the incentives for authors change from producing the most beautiful story to the most accurate one. And it's important to note that exploratory research is still possible, but it's distinguished from the confirmatory research. So you can still find interesting patterns that you'd like to explore later, but you don't pretend that's what you were looking for when you first started the study. Now, since the first journal in 2013, which was Cortex, some 200 journals have now adopted registered reports. These cover life and medical sciences, social sciences, physical sciences, and these include registered reports as an option. They're not compulsory. And Chambers does an assessment of where we've got to in these two reviews, one in Nature and one in the Journal of the Statistical Society, which are well worth a read. And they do seem to be working as intended. So the first analysis suggests that there's been a sharp rise in null findings. So in the general literature, we see that it's very rare, between only 5 and 20% of the time in papers do you find a null result. In registered reports for novel studies, that's gone up to 55%. For registered reports for replications, that's up to 66%. And there's a review there in PLOS coming to similar views. Looking within psychology specifically, it stays at 95% for the non-registered reports, but has gone down to that 40 or so % for registered reports. These are the proportions showing a null finding. And what's reassuring for the researchers is that these registered reports are being cited at a rate similar to non-registered reports. But what about early career researchers and open science in general, but also registered reports? And this has been looked at in a number of papers, but here are two reviews. And one of the points that both papers make is that although it may take more time, there is actually a significant advantage, which is that so far as your CV is concerned, you have a paper that you can go on your CV and you are guaranteed a publication, whether or not you find something that's super exciting. So long as the science is sound, you have a good publication. So there are a number of other more subtle effects, and those are discussed in those papers. So returning to power posing, what about the effects on risk-taking and hormones? So this is something that I think is really impressive. This is Dana Carney, the first author on that 2010 paper, has come to the view that the original study only showed the significant results because of p-hacking, as she realizes in retrospect. And she got involved in 7 replications using registered reports. The important conclusion was that none found the effect. In other words, there was no effect on risk-taking and hormones. There was a self-reported effect on how people were feeling, but the original study claimed that there was an effect on risk-taking and hormones. None was shown. These all came together in a special issue of a journal. And the important thing is that we can move on to study other interesting ideas instead. So there are a couple of other take-homes from the power posing story. And the first is that a positive literature can uniformly report an effect which does not exist. The meta-analysis can help, but it depends completely on the quality of the inputs. And it's a reminder that although physiological measures are very eye-catching, they're not necessarily conclusive. They can also suffer from small sample sizes. But the ambiguities were resolved by registered reports. So what about future human chemosensory research? I think ideally we ought to be using pre-registration when we can, and where it's possible, perhaps moving on to registered reports. One of the things about registered reports is that you get your peer review at the time when it's most useful, before you've collected the data. And again, open science would be the ideal. And I think to get those larger ends, we need to be looking at collaborative multi-center open science at large scale. And the GCCR is one example of those. There are so many more studies of human olfactory cues and the sense in general that we need to do. There's going to be a question generally about which of the classic observations are going to be replicable. And there are new technologies to measure odors. This is one from Craig Roberts in that symposium that came out in April. And just from a personal point of view, I do hope that we stop using these two molecules. Well, thanks for listening. I think we're going to have questions in chat, possibly by voice. If you'd like a copy of the slides, just send me an email, or they'll be going up on the web after this talk. And thanks very much again.
Thank you very much, Tristram, for a very nice talk. So we welcome questions from participants, and through the chat room, I think it's preferable. So Trist, if you can open the chat room.
Yep.
If you've got any questions, anybody, please type into the chat room.
Yeah, I'm not seeing the chat window. Oh, here we are. Right, got it.
Lots of very positive comments on the talk so far. No questions so far. So question from Simon. Simon Gain.
So do you think the incentives will align in such a way that we can manage to get to the open science utopia? I hope so. I think there's a new generation of scientists who will make it so, so long as they can survive. And I think that's something that's coming out very powerfully. There was a meeting of the King's College London Open Science Network, the Reproducibility Network, yesterday, and those talks are going online. And one of the strong messages was from early career researchers that they really need to have this coming in as soon as possible, and it needs people of my age to really foster the change. We've done well out of the system as it has evolved. We need now to support a new way of doing things. And notable was the presence of the Wellcome Trust as one of the major funders of research in this area, in psychology and medical research in general. And they're certainly looking at the ways that they could support this. I think it's also a long-term investment in your future. There's a good chance that the work that you produce in these new ways will be seen to be of greater value and is less likely to crash and burn. I think the overclaiming by researchers from very small studies can give you a very catchy study that goes in a journal with wide impact, that may turn out to be based on so few animals that it really isn't reliable. And I think we all love a story, but I think it may be better to take the time to produce something stronger. So yes, thank you.
Okay. So there's a question from Carl Philpott on the book Jacobson's Organ. Any comment on how that fits in?
So my feeling in the end is that humans don't have a Jacobson's organ. There's been quite a bit of dispute over the years. My understanding, I think, is it Tim Smith, the neuroanatomist, I think is one of the authors who's written most about this now, has come to the conclusion that it is present in the human embryo, but by the time we're born, it's completely disappeared in any functional sense. So I think the answer is we don't have a VNO, we don't have a Jacobson's organ. And along with the great apes, that's not something that we should be looking for. The important thing, though, is that we know from lots of other mammals, including sheep and goats and rabbits, that pheromones can be detected by the main olfactory system. That's also true in mice. And so there is no requirement for us to have a VNO or us to have pheromones. And I think that was actually one of the logical mistakes that was made back in 1991 when Monty Blanc and co-authors were producing their papers. The dogma was that to have a pheromone, you would need a VNO. That was what was coming out of the mouse research. And so in order to make their case convincing, they felt they had to do it through the VNO. And that's why they found it, even though it probably doesn't exist. That's why they were able to get recordings, even though it's highly unlikely that that was possible in any meaningful sense. So yes, it's a very interesting question, but I think the conclusion as of 2020 is that we don't have one.
Okay, thank you. There's a question from Lee Gibson. which looks very complex. What's your view on studies that have shown behavioral and fMRI responses to sniffing clothing, for example, ovulating women or aroused men?
I think there's a real problem with the way that there is huge researcher flexibility in the analysis of fMRI. And this comes from the fMRI researchers. There's a special issue of one of the main journals devoted to this whole topic of introducing registered reports to the journal. And there is a recent paper, I think published in the last month, where 70 researchers using the same set of data came up with completely different analyses of the same data. So in other words, there are so many ways to analyze fMRI that unless you say exactly how you're going to do it ahead of time, you can find all sorts of things. And I'm afraid that's also true of small behavioral studies. So I think it's highly likely that humans can detect the smells of ovulating given off by ovulating women. I'm not sure we have reliable evidence yet. So I think that's going to be the fascinating area as we go forward, that this will be a risky business. We almost ought to be looking at papers when we're citing them and thinking, okay, how likely is it that that paper would replicate? But that's a very hard thing to do. And it's the kind of thing that nobody would like to do. But yes, that's the kind of thing we ought to be doing. So the answer to the question is we simply don't know. There are indications it might be, but we simply don't know.
Okay. Yeah, from Bob Pellegrino, how do we prevent the incentives of money for open journals? For instance, the more they publish, the more they get paid by researchers.
Yeah. The hope of open access publishing with the author paying a contribution towards the cost of publication isn't working as intended. And what's happening is, as the questioner Bob implies, this now is a major source of revenue, and there's also double dipping by the traditional toll publishers, as we might call them, the Elsevier's and Nature Springers. It's a real problem. This week, MIT has failed to renew its subscription with Elsevier, and the reason was this was one of the problems that could not be solved. And I think the answer is to go one stage further. We're still caught up on impact factors, journal impact factors. And so you have the bizarre situation now that with the APCs, the author publication charges, for the implied kudos of simply appearing on a Nature platform, people are submitting to the Nature family, like Nature Human Behavior, rather than to the PLOS journals, because it comes from the Nature website URL when you click on it. And even though what it means now, you can pay more to submit to Nature Human Behavior than to a PLOS journal. And this will lead to all sorts of perverse incentives. And richer labs will be able to pay that extra premium. So I think we do have to move to a different model. What a number of researchers, I think it's the Cornell University Library System, has shown that the real cost of publishing papers is tiny, and that the value added by the traditional publishers is minuscule. And so what we need to move towards is a model of publishing that actually has realistic APCs, which are in the hundreds of dollars per publication only. And perhaps to finish the answer, there are a few examples of how that can be done. And one of those is in the range of publications that are coming out of some universities and some of the other smaller startups. And they have publications, APCs of a few $100s, and that's a break even. So that's where we need to go. Where we're likely to be going in the short term, unless there is a revolution, is the same big publishers maintaining their 30 to 40% profit margins, but just using it or gaining that money in a different way through the APCs rather than a library subscription. So the revolution needs to continue. Thank you.
Okay. The question from Anne-Sophie Barish. Could you explicate the connection between the original pheromone experiment and the question of replication? She said that what I found interesting is that the notion of pheromone here was operationally bound to an experimental protocol and dropped in later studies. What's happening here? Conceptual change? How could a greater focus on operational notions of central terms in psychology inform the broader issue of replication?
Ooh, I think the biggest problem is the attraction of conceptual replication. The original kinds of study that would meet the original operational criteria are really hard to do. And not everybody has 5 years or however long it would take to do those kinds of studies. In the context of olfaction and studies on the sense of smell, there's an extra problem, and that is that you need a really good chemist. But without a good chemist, you simply can't do the sorts of analyses and be aware of the chemicals that you're delivering in your experiments without running them through GC and so forth. And so in the absence of having a good collaboration with a chemist or being able to do those analyses yourself, you are driven to do something different, to do a secondary kind of research. And in the case of the human pheromone work, using those two molecules, androstadienone and estratetraenol, one of the attractions for psychologists was that you could do this in a lab with no chemistry background, with no chemist involved, simply by doing it by mail order. You could pay your money and get the molecules in the post and get to work. And so those kinds of experiments were very attractive, and there was every disincentive to doing them properly in the way that one might think. So I think you're right. When you're doing further research in a particular area, you do need to keep on going back to the foundation studies. The problem is that until now, with the changing editorial policies, where Royal Society Open Science, for example, will undertake to publish any replication that is well made, generally speaking, you'll be rejected at the first post on the grounds that this is not original and exciting research. So I think there needs to be a change in funding and a change in the gatekeeping at journals. So yes, very good question. Thank you.
Okay, right. We're not going to be able to cover all the questions that have come up, but there's one comment from Stuart Firestein. Has anyone done a review of a pre-registration submission for a journal that then agrees to publish a paper no matter how it turns out. He says, I'm curious about how one does this precisely. Are we going to wind up with a cadre of statisticians reviewing pre-registrations or papers?
Yes, there's been a lot of postmortems. So, and some of the postmortems are throwing up problems. So, for example, and there are references in my 2020 paper, people looking at the first cohorts of registered reports and finding a distressing number of cases where people didn't do what they said they'd do. Even worse, people didn't make available what they'd promised to do. And so we were kept in the dark as to what they'd promised. So there are a whole series of reforms that are coming in to make good practice as good as it could be. And so some of the commentaries are basically saying, okay, this was the ideal. Clever people continue to work around them. This is how we need to tighten them up further. But the answer is, yes, we are going to have statisticians getting much more involved with that review of studies. What it could do, though, is increase the quality of studies. And the good thing for the researchers is basic errors of missing controls or sample sizes too small might be caught before you do the experiment, not that gotcha, which basically torpedoes the experiment when it's too late and the grant money has run out. Can't do anything to do something about it. So hope is it might even improve things.
Okay, so thank you very much. I think we need to stop here. And I thank everybody for participating. And on behalf of the UK Chemosensory Network, thank you again, Tristram.
Great, a pleasure. Thank you.